All right, welcome. Today's mission, should you choose to accept it, is to absolutely master your GCSE practical on human reaction time. We're going to break it all down step by step so you can not only ace the experiment itself, but completely crush any exam questions that come your way. Ready? Let's dive in.
So here's our game plan. First, we'll get into the biology of it all. Then, we'll run through the practical itself, and we'll finish up with some absolute killer tips for your exam. I promise, by the end of this, you are going to have a complete toolkit for success.
First things first, let's get the fundamentals down. You see, to really understand this practical, we've got to understand the biology humming away under the surface. What are we actually measuring when we talk about reaction time?
I mean, just think about it for a second. Catching your phone before it hits the floor, slamming on the brakes in a car, even just swatting a fly. You are using your reaction time constantly, every single day. But what's really going on inside your body to make that split-second action possible?
Well, to put it simply, reaction time is just that tiny delay between a stimulus, like seeing that ruler start to fall, and you actually responding to it. And here's the key. It's not just about how fast your muscles are. No, it's a direct measure of how quickly your nervous system is firing off messages.
This pathway you see here, this is the entire journey of that reaction. It all starts with a stimulus. You see the ruler fall. That triggers an electrical impulse that zips from the receptor cells in your eye all the way to your central nervous system, your brain and spinal cord, for processing. Then a new message is sent out to the effector, in this case the muscles in your hand, which triggers the response. The time it takes for that whole trip, that's what you're measuring.
Okay, so we've got the theory down. Let's get practical. How do we actually design an investigation that's going to give us reliable results and, let's be honest, get us those top marks in the exam?
And here it is. This is your core objective, straight from the AQA specification. Now, notice that key phrase, the effect of a factor. This is so important. You're not just measuring reaction time in a vacuum. You're investigating how something, like having a coffee, being tired, or listening to music, actually changes it. That's where the real science is. So how on earth do we measure this lightning-fast process? With a fancy machine? Nope. We use a surprisingly simple but really effective technique. The good old ruler drop method. The method itself is really straightforward. Your partner holds the ruler, lets it go without any warning, and you catch it. The distance it falls before you grab it is your measurement. But listen, the most important step on this list is the last one. Repeat. You have to do this multiple times to get reliable data and to help you spot any weird results.
Okay, now we're getting to the bit that really separates good science from just messing about in a lab. To get valid conclusions, the kind an examiner is going to give you credit for, you absolutely have to control your variables.
So, there are three types of variables you need to get your head around. The independent variable, that's the one thing you deliberately change, like having a caffeinated drink or not. The dependent variable is what you measure, in this case, the distance the ruler drops. And then you have the control variables. That's everything else you must keep exactly the same to make sure it's a fair test, like using the same person, the same hand, and making sure there are no distractions.
Now listen up, because this is a massive exam tip. It is not enough to just list your control variables. You have to be able to explain why you're controlling them. For example, you control distractions because they could affect your concentration, which would unfairly change your reaction time. You always have to link the control variable back to its potential effect on your results.
Okay, these two words, they trip people up all the time, but the difference is actually really simple. Your results are repeatable if you can do the experiment again and get pretty much the same results. Your results are reproducible if someone else can follow your method and get similar results too. Both are super important for making sure your findings are trustworthy.
Right. You've done the experiment, you've collected all your data, the mission now enters its next phase, analysis. How do you turn a boring list of numbers into a clear scientific conclusion?
Okay, let's have a look at some sample data. We've got 5 trials for each condition, with caffeine and without. At a quick glance, the numbers for with caffeine look a bit lower, right? That suggests a faster reaction time. But take a closer look at that no caffeine column. See anything odd? Yep, there it is. That 28-centimeter result. It is way higher than all the other readings in that column. Maybe the person was distracted for a second, maybe they sneezed, who knows? In science, we call this an anomalous result, or an outlier.
So you found one. Now what? Do you keep it in your data? Do you pretend you didn't see it? Your next move here is absolutely critical for making sure your conclusion is a fair one.
And the correct move is you ignore it when you're calculating your average or your mean.
If you included that 28, it would pull your average way up and give you a misleading result. It was clearly a poor measurement, so we exclude it to keep our data accurate.
And once we take out that anomaly and we calculate our new means, the pattern becomes crystal clear. With an average of 15 centimeters for no caffeine and 11 for with caffeine, our bar chart shows a really obvious conclusion. In this investigation, caffeine seemed to make reaction times faster.
You've done the practical. You've analyzed the data. Now, for the final part of the mission, using all this knowledge to lock in those top marks when you're sitting in that exam hall.
Look, in the exam, they won't just ask you to describe what you did. They're going to ask you to evaluate it. This is where you show you can think like a real scientist. You need to ask yourself these kinds of questions. Could you have improved reliability by doing more repeats? Was there a chance of a systematic error creeping in? Could a computer test have been more precise? Answering these kinds of things is how you get to those top-level marks.
And I'll leave you with a final thought to chew on. You see, great scientists don't just follow methods, they adapt them. You now have a solid framework for investigating a factor's effect on reaction time. So, how could you maybe adapt this kind of method to investigate something else, like the effect of practice on memory? Thinking like that, that's when you know you've truly mastered the science. Good luck.